Sound. Sound has enormous power. This story is about the birth of a new direction in medicine, vibrational acoustic homeopathy. We are all made up of vibrations. Our world and our universe is music. In the beginning was the Word, the first line of the Gospel of John, New Testament. Each molecule has its own unique molecular weight and creates its own unique vibrational music. Just like the cells of our body, each organ in our body plays its own unique vibrational music. Having learned to correctly read this vibrational musical matrix, we will be able to influence our body and even learn how to program stem cells non-invasively. Today, I will introduce you to unique research results made by various groups of independent researchers around the world. Programming stem cells through a sound matrix. Researchers used laser equipment to count the vibration matrix from living cells of the nascent heart, from which the vibrational musical matrix was initially read. Next, this acoustic matrix was fed through ordinary speakers to the stem cell culture, and after 2 weeks, they turned into heart cells. In those cells, from which the vibrational musical matrix was initially read. This is a human heart cell. Contractions and pulsations, this laser sensor records in the form of drawings. This is a sound vibration code. We can listen to the sound created by this frequency. When this sound was played near undifferentiated human stem cells, 2 weeks later they became pulsating cardiac muscle cells. These are cardiac cardio cells taken from human stem cells. They have become absolutely identical to the myocardial cells with the original ones for that sound vibration. And so each cell and biomacromolecule has its own unique sound code. Non-invasive regeneration of pancreatic cells in rats. Research group of Petr Gariaev. Any substance that is present there in the laser beam, information is read from it. Here is a piece of quartz. I insert it under the laser beam. I expose and adjust the laser beam in a certain mode and we get sound generation. Now there is a clear signal, so I block the laser beam and the sound disappears. I open and generation returns. What is the result of sound? Is this how information is transmitted? Yes, yes. In this case, if there was not a quartz crystal, but a blood sample, we would receive a transfer of this information to a person. If the person is a relative of the donor of this blood, he receives corrective information, healthy blood of a newborn child, and the recipient is ill. It corrects his health and even slows down his aging. A group of researchers used laser equipment to read a vibration wave matrix from a live culture of pancreatic cells taken from newborn rat pups. Next, this vibration audio matrix containing information about the healthy state of pancreatic cells is applied. They exposed sick adult rats with damaged pancreas to this audio matrix. After a week of the experiment, most of the rats recovered. After autopsy of the rats, regeneration of the damaged pancreas was discovered. DNA teleportation experiment. Luc Montagnier's research group. In his experiments, Luc Montagnier captured a vibrational electromagnetic field from DNA molecules or blood cells. By digitizing the resulting spectra, they turned into audio files similar to matrices containing electromagnetic vibration information about cells. Then, through a conventional audio amplifier, An electromagnetic audio matrix read from DNA samples with a given sequence was supplied to a solenoid with a test tube of pure distilled water. And thus, they managed to conduct an experiment on DNA teleportation in a non-invasive way. Blood music. John Stuart Reid.
It's John Stuart Reid, and I'm an acoustic physics researcher. Pythagoras said that music could be used in place of medicine. It got my attention, and I thought there must be some experiments that could be conducted where we can carefully measure the response of music to some sort of healing situation. The first experiment that I conceived was using blood. You take a vial of blood and you place it into an incubator with a small loudspeaker in the incubator that's feeding music to that blood. At the same time that that's happening, we have another vial of the same blood. We have Faraday cage, which is electromagnetically screened, but it's also a very, very quiet environment. So that blood is not receiving any music, it's just receiving quiet. We go through a protocol that first dilutes the blood by a certain ratio with a buffer solution, and then we extract a small amount of that mixed blood. When we put that blood into the cell counter, it will tell us how many cells are actually live and what the term is viable. The cells in the Faraday cage have not fared very well at all. Mostly dead.
But if the cells are dead, that means that the cells from the tumor got exposed to was it's the same thing.
I think we should probably repeat that though and see if we get the same result. Still the same. Dilute the sample, more cells alive than the cell count can count. That's a huge difference between the music and the quiet. Well, look.
This is amazing.
So I think we should repeat it, see if we get the same result. Above the range that it can count, cells that are transiting towards being dead, but being actually really alive to the extent that so many of them are now alive. It's really wonderful. It's an amazing result.
ADARIS Laboratory. Development of methods and equipment for vibration medicine. We develop equipment capable of recording the unique vibrational music of cells or biomacromolecules. In 2023, we were able to recreate equipment capable of recording the smallest nanovibrations of biomacromolecules and translating this information into sound in real time. This makes it possible to create various nanovibration matrices from various drugs. For example, what do trehalose molecules sound like in an aquatic environment? This is the Trehalose vibration matrix created in the ADARIS laboratory. The second direction in our developments is improvements, MBER laser spectrometer, for creating so-called Gariaev matrices from various biomacromolecules. Success was achieved in obtaining very pure and high-quality matrices using Gariaev's technology. A user-friendly software allows you to visualize the recorded spectra in real time, which simplifies laser setup and the creation of matrices from various drugs or chewing cells. ADARIS Laboratory, we are changing the world.